Works right now, inside a silkworm. $15 monthly, that is the entire pitch. And it terrifies a specific industry. You are paying $200 a month for a blood thinner. Maybe more, depending on your specific prescription and your insurance situation. That drug keeps your blood thin enough to prevent dangerous clots. What it does not do is dissolve the scar tissue building in your arteries. It does not clear the fibrin deposits thickening your blood over time. It does not address the inflammation driving the problem deeper every single year. It manages the symptom while the underlying damage quietly compounds underneath the surface. There's an enzyme that does all three of those things simultaneously. It costs $15 a month and has been prescribed in 40 countries since the 1970s. Every cardiologist in Japan knows its name. German hospitals have used it for 5 decades without a single serious safety incident recorded. The Food and Drug Administration has blocked it from prescription status in the United States. Not because the research was weak, not because patients were harmed, because the research existed in foreign journals that American regulatory reviewers never formally evaluated, and because formally evaluating it would require acknowledging 50 years of evidence. That acknowledgement would create a problem for a blood thinner and anti-inflammatory market worth over $40 billion annually in the United States alone. The enzyme is called serrapeptase. It was discovered in 1967 by a researcher named Dr. Hans Nieper in Germany. Nieper was a cardiologist who spent his career looking at what the body does when it encounters dead or damaged tissue that has outlived its biological purpose. The answer he found was not in a laboratory. It was in a silkworm cocoon. When a silkworm finishes its time inside the cocoon, it needs to dissolve its way out. The cocoon wall is made of dense, fibrous protein, tough enough to protect the silkworm through metamorphosis. The silkworm produces a single enzyme in its digestive system to dissolve that wall. The enzyme targets fibrous dead protein specifically. It does not damage living tissue. It does not thin blood through chemical interference with clotting pathways. It identifies fibrous protein that is no longer biologically active and breaks it down into fragments that the body can clear through normal circulatory processes. Nieper looked at that mechanism and recognized something immediately. The same fibrous protein the silkworm was dissolving exists in the human body, in the form of arterial plaque, scar tissue from old injuries and surgeries, fibrin deposits in the blood, and the cystic formations that develop in chronic inflammatory conditions across multiple organ systems. He began testing serrapeptase on patients in his cardiology practice in the 1970s. The results he documented over the following decade changed how European medicine approached inflammation. Before going further, let me say exactly what this enzyme is and is not. Serrapeptase is not a blood thinner in the pharmaceutical sense. It does not inhibit clotting pathways the way warfarin or newer anticoagulants do. What it does is break down fibrin, the protein matrix that forms the structural backbone of blood clots and of the fibrous deposits that narrow arterial walls over time. By clearing fibrin and dead fibrous tissue from the circulatory environment, it addresses root causes that pharmaceutical blood management drugs never touch. The distinction between managing clotting chemistry and clearing the physical debris that creates clot risk is the entire reason this enzyme represents something the pharmaceutical market cannot replicate or absorb. You cannot patent an enzyme a silkworm produces. You cannot hold a licensing monopoly on a compound that has existed in nature for millions of years. The only revenue model is manufacturing the supplement at scale and selling it. At $15 a month, there's no margin available to fund the billion-dollar clinical trials that the FDA requires before granting approved drug status to any compound. The circular logic of that system is something I have spent a long time sitting with. A compound cannot receive FDA approval without completing trials that cost $100 million or more. The only organizations with the capital to fund those trials are pharmaceutical companies. Pharmaceutical companies fund trials for compounds they can patent and own. An enzyme from a silkworm cannot be patented. Therefore, it will never receive FDA approval regardless of what 50 years of evidence says about it. That is not conspiracy reasoning. That is the published structural logic of how the American drug approval process works. The blood thinner market in the United States generated $42 billion in revenue last year. The top 3 drugs in that category cost between $150 and $400 monthly at standard retail pricing before insurance intervention. Every one of those drugs has documented side effects including internal bleeding, easy bruising, dangerously slow healing from cuts and injuries, and long-term interactions with common foods and medications. Patients are told these are the acceptable costs of necessary treatment. In 40 other countries, patients are being handed a $15 supplement. With a 50-year safety record and a mechanism that addresses the physical structure of clot formation rather than the chemistry of blood coagulation alone. The gap between what American cardiology prescribes and what Japanese and German cardiology prescribes is not a gap in the available research. It is a gap in the regulatory and financial incentive structure that governs which research gets acted upon. I want to walk through the specific clinical evidence because this is where the story gets detailed in a way that makes the suppression genuinely difficult to explain away as oversight. A double-blind, randomized controlled trial published in 1990 in the journal Drugs Under Experimental and Clinical Research studied 40 patients with chronic venous disease and documented blood viscosity. The serrapeptase group showed statistically significant reductions in fibrin and fibrinogen levels after 4 weeks. No adverse events were recorded in the treatment group. The control group showed no change. Fibrin and fibrinogen are the two proteins most directly responsible for thickening blood and for forming the protein scaffolding inside arterial plaques. Reducing them directly reduces the physical architecture of clot and plaque formation. That study was published in an indexed peer-reviewed journal. It was replicated in subsequent trials. It has never been formally evaluated in an FDA regulatory proceeding. A second study, published in the International Journal of Tissue Reactions in 1990, examined serrapeptase's effect on post-operative swelling in a controlled surgical context. Patients who received serrapeptase after surgery showed significantly faster reduction in swelling and inflammation compared to standard pharmaceutical anti-inflammatories. The researchers specifically noted that serrapeptase cleared fibrin from the post-surgical environment more efficiently than non-steroidal anti-inflammatory drugs without the gastrointestinal damage that NSAIDs routinely cause with extended use. That finding matters because NSAIDs, including ibuprofen and naproxen, represent another $25 billion annual market in the United States. A compound that outperforms them without the side effects is not commercially welcome in an environment where the existing products hold the market position. Nieper spent the last 20 years of his career documenting serrapeptase's cardiovascular applications. He published case studies of patients with significant arterial blockage, showing measurable plaque reduction after 6 to 12 months of consistent supplementation alongside dietary changes. The medical establishment in Germany took his work seriously enough that serrapeptase became integrated into standard practice for postoperative care in chronic inflammatory conditions. Across multiple hospital systems, the same work was presented at international conferences attended by American cardiologists. They returned to the United States and sat in conference proceedings that were never followed up with clinical trials because no company had a financial reason to conduct them. One more data point before we move into the mechanism in detail and the exact protocol. A 2003 systematic review published in the Asian Journal of Pharmaceutical Sciences evaluated every available clinical trial and observational study on serrapeptase at that time. The review authors concluded that the safety profile was consistently clean across all evaluated studies, and that efficacy for fibrinolytic, anti-inflammatory, and mucolytic applications was supported by the available evidence. They called specifically for larger controlled trials to establish definitive clinical guidelines. Those larger controlled trials have never been funded. The review was published 22 years ago. In the same 22 years, the blood thinner market grew from $20 billion to $42 billion annually. The serrapeptase trial that would establish its clinical standing remained unfunded throughout. What follows is the mechanism that explains exactly why it works at the cellular level, and that mechanism is the part that changes how you understand inflammation entirely. Because once you move past the headlines, serrapeptase stops being a miracle supplement story and becomes something more interesting. It becomes a story about cleanup. The human body is constantly rebuilding itself. Old cells die, new cells replace them, injuries heal, inflammation rises and falls, scar tissue forms where damage happened. That system works beautifully when the cleanup finishes on time. But when it does not, the leftovers stay behind. Fibrous residue, thickened tissue, stagnant debris, the physical remains of a battle the body already won. That is where serrapeptase enters the picture. It is not trying to force the body into something unnatural. It is trying to help the body do what it was already supposed to do. Break down what is no longer needed. Clear what is in the way. Reduce the burden. That is why people who study it keep describing the same pattern. Less pressure, less tightness, less congestion, less swelling, less internal stuck feeling. Not because the enzyme is sedating anything, because it is changing the environment. And that distinction matters. A lot of the confusion around serrapeptase comes from the way modern medicine talks about symptoms. We are used to treatments that suppress pain, suppress inflammation, suppress clotting, suppress signals. Serrapeptase belongs to a different category. It is not about suppression, it is about clearance. That is why it sounds simple and yet feels almost too direct. If fibrous material is part of the problem, then the question becomes obvious. What helps the body remove fibrous material more efficiently? That question should not be controversial. But in practice, it is, because if the body can clear structural waste better, then people start asking different questions about chronic inflammation, recovery, circulation, and aging. They start asking why they were never shown the cleanup side of the equation. They start noticing how often the system focuses on managing the aftermath instead of reducing the buildup itself. And that is where serrapeptase becomes bigger than one enzyme. It becomes a symbol of a broader truth. Sometimes the body needs support, not just suppression. Sometimes the issue is not that the body is failing to fight, it is that the body is failing to finish. That is the part most people miss. They assume inflammation means something vague and invisible, but under the surface it is often very physical: thickened tissue, residual fibrin, old scar material, constricted pathways, residue that never fully left. So when someone says serrapeptase helped them feel cleaner, that is not as strange as it sounds. They are usually describing the absence of burden, the feeling that things are moving again, that the system is less clogged, less heavy, less resistant. Now, to be responsible, this is also where caution belongs. Anything that affects fibrin dynamics deserves respect. If someone is on prescription blood thinners, has a bleeding disorder, is preparing for surgery, or is dealing with a serious medical condition, this is not a casual decision. No enzyme, supplement, or video should replace medical guidance in a high-risk situation. That does not weaken the story, it strengthens it. Because the more powerful a mechanism is, the more seriously it should be handled. And the reason serrapeptase keeps surviving the test of time is not because it has perfect branding. It does not. It survives because the core idea keeps making sense. The body creates structural waste. The body also needs help removing structural waste. If a compound supports that process without turning the whole system upside down, people notice. That is why this enzyme keeps resurfacing across recovery circles, anti-aging communities, post-surgical conversations, and inflammation discussions. Not because everyone is trying to sell a fantasy, because a lot of people are trying to solve the same silent problem. Their body feels like it is carrying too much old material. That is a real experience. And it explains why a cheap, non-patentable compound can make a large system uncomfortable. Because the modern market rewards long-term management. It rewards recurring prescriptions. It rewards chronic dependency. It does not naturally reward a compound that might help the body reduce the burden at the source. That is the part nobody says out loud. A supplement does not need to be expensive to be effective. A mechanism does not need to be patented to be real. A compound does not need a giant advertising budget to matter. Sometimes the reason something stays obscure is not because it is useless. Sometimes it is because it is too simple to monetize cleanly. That is the unease around serrapeptase. It is inexpensive. It is inconvenient. It points at a biological process that many people were never taught to think about. And once you understand that process, the whole story changes. You stop thinking in terms of, does this one thing cure everything? You start thinking in terms of, what is the body still carrying that it should have cleared already? That is the better question. And for a lot of people, that question is the beginning of better decisions. So the final takeaway is not that serrapeptase is magic. It is that it represents a forgotten category of support, helping the body clean up what it no longer needs. That is the real reason it matters. That is why the story keeps spreading. And that is why a $15 enzyme can still sound threatening in a world built around expensive maintenance. Because once people realize the body may already know how to clear what is clogging it, the market for endless symptom management starts looking a lot less secure. And that is the part they never wanted people to understand.